introduce Yoland. Yoland is, I think, your primary appointment's in neurology. Am I remembering this correctly? He's professor of neurology at Emory and also a division chief at the Yerkes National Primate Center there. I'm going to embarrass Yoland by saying I think he is really one of the very best neuroanatomists on the planet. He has a very distinguished pedigree. He was a graduate student with Andre Parent in his native Quebec, and then did postdoctoral training with J.P. Bolam, and has gone on to do a very large volume of really impressive and frequently quite beautiful work on non-human primates. So he's going to talk to us today about some of the recent work that he and his colleagues done recently, largely relevant to the activities at the Udall Center at Emory. All right, Yoland, thanks very much.
Okay. So thanks a lot, Roger. It's very nice introduction. Thank you. It's too kind of you. And thanks a lot for the invitation. So it's really also a great pleasure to talk to you. Of course, we know a lot about your team there and University of Michigan when it comes to time to talk about basal ganglia. And I mean, this is definitely a place where a lot of great stuff is doing. So hopefully you'll enjoy what we'll talk about. And so let me just share my screen and we'll get going. Just see here. Okay, here we go. Share this. Slideshow. Okay, good. And I try to. Second here. I'm just trying to remove. A second. Okay, here we go. Perfect. Okay, good. So, so again, thanks a lot for the invitation. So it's a great pleasure for me to be, and hopefully you will be able to chat a little bit about what we're doing here. It's not, of course, only myself, as I think, as Roger mentioned, we have a team. We have a Udall Center here as well. So we have a team that is working on Parkinson's disease, really related to the pathophysiology. This has been the main kind of contribution that our group and dates back essentially from Malin Long and others and Thomas Wickman and others. So this is really an understanding essentially various aspects of the pathophysiology of Parkinsonism and using non-human primate as a model. So this is what I want to do in this talk. In fact, it's mainly to update you. It's really work in progress. So the fact that there will not be a lot of new published data. I think this is because this is a research program that we have started quite recently, and you will see what we're interested in terms of the cortical output and changes in cortical activity in monkey model of Parkinson's disease. So we'll just really get going on that. So I would present some of these preliminary data and the techniques that we're using and the goals of this, and I definitely welcome inputs and suggestions or comments or things that you find really do not make too much sense. But I think that would be great to hear your thoughts about that as we kind of move this project forwards. So as Roger mentioned, all this work is done at the Emory National Primate Research Center, and this is where our lab is located, which is shown. Just put my pointer here. So that's the front entrance of the Primate Center, and this is the Emory campus, and this is our team here, which is really, we have this ASAP grant as well. So that's our ASAP team. So we had a meeting with the different group here a few weeks ago, and this is a picture taken from the team here. Okay, so the main, as you know, I think you don't need any introduction of this model, particularly coming from Michigan, and with Roger there, who is really one of the who introduced really the original version of that model. So this box and arrow model, of course, as you know, has been instrumental in the development of our understanding of the basal ganglia function and basal ganglia pathophysiology in Parkinsonism. So there's been a lot of work done, of course, at many levels, using anatomical, functional, imaging techniques, and so on. One thing that has been neglected, and I think is still relatively poorly understood, is what really goes on in the cerebral cortex in Parkinsonism or in Parkinson's disease in general. So clearly the two areas, the cortex And the thalamus is also another area. So today we'll talk mainly, we have been doing some work on the thalamus as well, but today I will focus on the work that we're doing on the cortex and try to really give you an ideas about why we believe it's important and how we're approaching it in terms of studying the pathophysiology of cortical outflow in these non-human primates. So in term, personally, as I think as Roger mentioned, I'm a neuroanatomist, so I've been studying, functional neuroanatomy of the basal ganglia and changes, neuroplastic changes in that circuitry associated with Parkinsonism. So I will be mainly focused today, my talk on the anatomical component of our project. This is, of course, there are also, I will let you know a little bit about what others are doing so that you see it's a kind of a composite of anatomy and electrophysiology that we try to pull together to understand that. But the talk is going to be mainly focused on the anatomical component of our project. So what I would try to elucidate into this is the question at the anatomical level is how is the morphology and the synaptic connectivity of corticofugal neurons are altered in Parkinsonism. And I put corticofugal neurons here because we will be studying various population of cortical output because we believe that this is important and this is something which has been neglected over the years. Of course, we always think about, pyramidal tract neurons, and then we think that all these cells that are going down the entomolar capsule and to the brainstem essentially are all part of the same bucket. And I think that's of course, like anything else, it's oversimplistic and it's much more complex than that. And there's many more population of neurons that are giving rise to output of the cortex. And I think we should start. I think we have the tools now to start interrogating these different subsets very specifically, and I think we'll gain a lot in being more careful in trying to really target very specific subset of neuron based on their anatomical connection. So the projects that we have in place, essentially, there are at the anatomical level, there are three main goals that we try to determine through this the Udall and this ASAP project is, first of all, we are interested in assessing pathological changes in the morphology and the connections of these corticofugal neurons. We'll be studying that mainly in the M1, the primary motor cortex, and also the supplementary motor area of these chronically treated MPTP Parkinsonian monkeys. We're also interested, and that's an important aspect that we'll talk about, is to look not only at the neuron themselves in the cortex, but also looking at their connections. So looking at their efferent connectome here. So we have tools that we believe are able to trace all efferent connections of individual cells in the cortex. So we'll be taking advantage of that to study changes in this efferent connectome of this subpopulation of corticofugal neuron in Parkinson monkeys. And that is mainly the ASAP project here, which is essentially to look at changes that happen into these morphological changes in neurons and also the output during the course of dopamine depletion. So essentially looking at that at different time points following chronic dopamine denervation induced by low dose of MPTP. So essentially looking at this not only at the end stage when the animal is Parkinsonian, but looking at that when the dopamine goes away to try to better understand whether or not some of these neuroplastic changes are associated directly with the development of Parkinsonian motor symptoms or other type of symptoms, or they are mainly ongoing homeostatic changes going on during the progressive loss of dopamine. So we'll be focusing on both M1 and SMA. So in primates, the motor cortex, the M1 is relatively large, but also there is very large other areas like the premotor cortex and the supplementary motor areas are very large areas that are, in fact, the SMA is the main target of the basal ganglia output from the thalamus. So there is a very strong projection to the SMA. There is also some going to M1, but definitely the SMA is the prime target of the basal ganglia outflow, if you want. And so we'll be focusing on these two regions. Both of them are sending projection to the spinal cord in primates and pretty significant projection. Of course, M1 is very strong. SMA is a bit lighter, but still going directly to the the spinal cord. And of course, both of them are heavily connected with the basal ganglia. So the idea, essentially very simplistic, is that really clearly the loss of dopamine, the loss of striatal dopamine, but also the loss of cortical dopamine, which we're not going to talk about today, but that's something also important to keep in mind, that there is a pretty significant amount of dopamine locally in the motor cortices of primates that we know very little about the role of this dopamine, this local dopamine. That's something that definitely need to be paid attention to. But the loss of dopamine is going to affect, the output of these neurons, and then we're going to really look at that. There has been some studies, in fact, that have been done, and I will talk to them about them as well by other group, essentially showing some changes in bursting activity of corticospinal neurons in Parkinsonian animals, and also changes in firing rate. What is not known is the temporal development of those changes. When does that happen? And also whether or not, how much of that involved the SMA, which has not been really looked at very much into these studies. So as a background, as a rationale for some of the work that we're doing, is some work that we have recently that was published last year into cerebral cortex. So one thing we're interested in here is to look at the thalamic innervations of motor cortices in primate in control and Parkinsonian monkeys. So what we use to do that, so we use an antibody raised against the vesicular glutamate transporter 2, which in that case, in the cortex, is a good marker of thalamocortical terminal, because the thalamic neuron express VGLUT2. There's the cortical neuron themselves express VGLUT1, so there's no VGLUT2 in the collaterals of the pyramidal cell neuron. So the VGLUT2 terminals in the cortex are really coming from the thalamus. And so when you look at that, in fact, you can see here all the little kind of white red dots are these axon terminal that spread across, different layers of cortex. And in primate, you clearly have some major input into layer 2, 3, and also some in layer 5. So what happened in the Parkinsonian monkey, so here is a fully Parkinsonian monkey, you have a significant decrease in the innervation of the deep layer of the motor cortex. You find that in M1, and we also found that in SMA as well. So there is some kind of reduced, at least, expression of VGLUT2. So that's something which is, we don't know exactly yet what is going on, but there's definitely a reduced immunostaining for VGLUT2 in the deep layer of the motor cortex. Another change that happened as well in M1 is that some neurons, subset of neurons, and these are preliminary data. This is not published. This is something that we're going to study in great detail in our project, is that there is a loss of spine on the dendritic tree, the apical dendrite of some of pyramidal cell in the M1 cortex in MPT treated monkeys. And you can see that pretty obviously here on these dendrites. You see a control dendrite, and here is an MPT treated monkey. So pretty significant innervation. And you see some quantification here. It's a relatively small number of pyramidal cells, but this was pretty obvious that there were some neurons that were really strikingly affected. And it seems to be somehow quite specific to M1. I think we have not looked at SMA in that case, but if you look at the cingulate cortex here, which is very close to the M1, then you do not find that. So there seemed to be something going on quite specifically in the motor cortex, at least in the based on these data. So the idea is that whether or not, the question is here, we saw that there are these changes in innervation, these changes in morphology. The question is, what type of neurons are affected by that? So do these changes affect specifically a population of corticofugal neurons, or is it a broad range and really any pyramidal tract neuron is going to be affected? And also how these changes affect the synaptic connectivity of specific population of corticofugal neurons in Parkinsonism. So these are the kind of the gaps that we hope to be able to elucidate into this project. And these data here are some data coming. This is not from our lab. This is a colleague of us, really, Young Chu, that probably some of you know well because he's very has been doing some great work in the basal ganglia, which is at the Venendel Institute. So what Ong-Yon found in studying this system as well in rodent model of Parkinsonism. So he's using, in that case, he's using the 6-HGA treated mouse model of PD. And he's interested in looking using slice work. He's interested in looking at the effect essentially of thalamic stimulation using optogenetics. So you can see here, he put, the channel rhodopsin in the VM, which is the main output of the thalamus in rodents that give rise to a lot of labeling of terminals in the motor cortex, and then he stimulate that, and he record the activity in cortical neuron, the response induced by thalamic stimulation. And what he found different thing, and which is interesting. First of all, he found that there was a decrease in the amplitude of the EPSC when he in the pyramidal tract neuron compared to the intratelencephalic cortical neuron, which is the other population of pyramidal cell that project only in the telencephalon. So we found, and you can see that there is a pretty significant decrease in the amplitude of those EPSCs, and there's no change here when you record from IT neuron. So indicating that there is some kind of decreased impact of the thalamocortical system on the pyramidal tract cells. The other thing that he showed as well, which is really very in line with what I just showed you in the monkeys, he also showed that there was a decreased density of VGLUT2 terminals in the M1 of the 6-OHGA treated rat. You can see that here, the number of varicosities of dots, essentially, which are thalamic terminals, is lower in the 6-OHGA treated animal than it is in the control, and you see the quantification of that here. So also in this case, a decreased VGLUT2 innervation of the motor cortex in 6-OHGA treated animals. And finally, he also found that some neurons are really undergo a loss of spine into the layer 5 of the M1. And you can see that here really being quantified. So you see the density of spine, onto these cells going down in the 6-OHGA treated animal. So there seemed to be, then it looked like there is, following the, using 2 different animal models, and which lead to mainly dopamine denervation, but also maybe in the MPTP, maybe other neurotransmitters. So it leads, to this kind of really changes into the M1. So I know that Ong-Yon is studying that as well in the M2 region of the cortex. So this is one part of he's part of our ASAP project, and he's looking at the M2 region as well. So the question is, what is going on there and why, how that happened? And this is something that I know that Ong-Yon is going to study that. So I'll try to understand the mechanistic underlying substrate of these changes, which we do not really know. One thing that came to mind for us, at least in the MPTP treated monkeys, is whether this loss of VGLUT2, some of the lesion or some of the degeneration of the caudal intralaminar nuclei in the MPTP treated monkey might contribute to that. It's not, we don't have any direct evidence for it, but just want to share with you a few data to indicate that maybe there's something that may be contributing to this here. So first of all, before showing this data, I think it's an important point to make, that there is a loss of thalamic neurons in Parkinson's disease, and this has been shown by neuropathological study by the group of Glenda Halliday some years ago now, and this, but on the other hand, this is quite restricted. So it's not in the ventral motor thalamic nuclei. It seems to affect mainly the caudal intralaminar nuclei, including the central median and the parafascicular nucleus. And you can see here that this is the nuclei. This is in human PD patient here that she did this work. And you can see the changes in the number of neuron in the CMPF, of Parkinson's disease patient. So there is definitely a pretty significant loss of cell in those nuclei. The other thing which was interesting that Glenda demonstrated is that it looked like the extent of cell loss is not directly linked with the severity of the Parkinsonian motor symptom. And this is what you can see here, essentially, on a your scale, you can see the stage 2 and 3 or stage 4, 5, you have the percentage already at the stage 2, 3, already the CM, the number of neurons that they generate in the CMPF has already reached its plateau. So it does not get, more, it does not get higher, in the very severely affected Parkinsonian patient. But there's definitely really a loss of neuron in the brain of people with Parkinson's disease in those nuclei. A few years ago, we showed that really you can mimic that, at least in part, using a chronically treated MPTP treated monkeys. So these are monkeys that are treated with low dose of MPTP for over many, many months. And then, gradually they will develop eventually Parkinsonism, but it will take really a lot of time. So, but they lose their dopamine over a long period of time. And in these animals, you find a pretty significant loss of neurons in the CMPF as well. And this is what is shown here. This is what you can see. We lost, pretty significant decrease in the number of neurons in the CM of this animal. And you can also see that, interestingly, if you treat the animal, with MPTP, but you do not let them become Parkinsonian. So that means that you sacrifice the animal after they have lost, let's say, 40–50% dopamine in the striatum, and already at that stage they have already lost, their CMPF neuron. So it seems like this is a process which start really pretty early during the in the degenerative process associated at least with the MPTP-induced, toxicity here. So to link that with the story that we had before is that why, that would be important. I think there is this nice data from André Parent here that was published, some time ago now. But what they did here is that they were studying by doing single cell feeling in monkeys, feeling individual neuron in the central median nucleus and following their axon trajectory and their projection to the striatum and also to the cerebral cortex. And what they reported in this study is that the CM neuron, they had quite a few of them that project to the cerebral cortex. These neurons terminate exclusively in the deep layer of the cortex. Okay, they terminate in layer 5 and maybe layer 6, but they go really deeper. They don't reach the superficial layer, which is very different from the projection coming from the ventral motor nuclei, like the VA, the VL, and so on. And you can see here, which is shown in the paper that Susan Haber traced and did really some years ago. So those, of course, are going to the superficial layers and also the deep layer, but clearly there is a massive projection going into the layer 2 and layer 3. This is where the bulk of the input is going. So in that regard, essentially here, the CM innervation of the motor cortex seems to target the deep layer. So possibly the loss of these neurons, might maybe lead to a decrease of VGLUT2 immunostaining in the deep layer of cortex, in these monkeys. But we don't have the direct evidence for that, but that's something that we need to keep in mind as we move along. And I know that Ongyan this is something also that he's studying, and of course he can do that using slice work, so we can look at mechanistic study to try to understand that. And he has some data suggesting that there is a contribution of the NMDA receptor, at least in his model, associated with the VM input. There is some contribution of the NMDA receptor that might contribute to the synaptic pruning in this animal model. It's still something which is ongoing. There's no really a lot of data yet, but there seems to be on a path to have at least some kind of mechanistic hypothesis here that he can put forward for the reason why these terminals really are pruned and the spine are pruned in the state of Parkinsonism. Okay, so the question, so I think, as I mentioned at the beginning, one thing which we hope to be able to really advance here compared to what is already in the literature is to try to pay attention to the to specific subtype of cortical fugal neuron. Instead of looking at that as a whole, as a single population, we'll try to study different subtype of them. And the three subtype that we are planning to look at are the cortical spinal neuron going directly to the spinal cord, cortical medullary cell that go to the lower medulla in the reticular formation, and the cortical striatal neuron that give, some of those that give collateral, to the brainstem. So we'll try to study these three population of cells in those in our work. So as a background for what is going on in the cortex, here are two sets of data, which I think has been for us a foundation for pushing that, and I think paying attention to the diverse population of pyramidal tract neuron. Of this data, one set of data come from Rob Turner, who is in Pittsburgh. So Rob, what he did, this was done really some time ago in 2011. He published that with Pasquero here. So what they found in this study is that they demonstrated that they looked at either the cortical striatal neurons and the cortical fugal cell. I think put cortical spinal here, which is not really true. So these are, I think, stimulated. This was by antidromic stimulation. This his electrode was really in the pons, in the lower pons, essentially. So these are really cortical pontine neuron, if you want, that he versus cortical striatal. So what he found in this study is that these are, and he looked at that in the MPTP treated monkeys and the control, and he found that the firing rate of cortical striatal neuron was not affected in Parkinson monkey. You can see here the pre and the post MPTP for the cortical striatal neuron, more or less the same thing. On the other hand, for the cortical pontine cell, there was a significant decrease in the firing rate of these neurons when the animal was Parkinsonian. And you can see here the gray bar shifting to the left compared to the black bar, which are more really have a higher firing rate. So here suggesting that cortical striatal and this cortical pontine neuron form two distinct populations that are affected differently in Parkinsonism. Other data that were published by Rob, look at that as well, but also here from Joshua Goldberg, and is the aspect of increased synchronization or bursting between neighboring M1 neuron in Parkinson monkeys. And here I'm just showing just an example of that. So if you have in a normal monkey, then most neurons really next to each other will not be synchronized. At least the synchrony will not be very high in terms of bursting. However, when the animal becomes Parkinsonian, then you have these neurons, which is a typical feature of in Parkinson, not only for the cortex, but many other regions, but the neuron, really the start bursting, in synchrony. So which is something which was noticed as well. So there are those changes going on in terms of firing rate, in terms of firing pattern, of synchronization that is going on in the cortex, but there's still a lot to know about what may be contributing to these changes. So I will just here go over one study that we're trying to approach that problem, the way that we're approaching that, at least in the in our Udall project using rhesus monkeys. So here is kind of the design that with the tool that we are using to label that. So we're focusing here on the cortical spinal neuron. So neurons that are mainly really going to the arm area of the spinal cord. So we're using group of rhesus monkeys. 6 monkeys at least will be controlled. This is mainly for the anatomical study with 6 monkeys in the control group, 6 that will be treated with MPTP. So the way that we're going to label those neurons is to place injections of an AEV, which is a retrograde AEV that we put into the spinal cord that will label retrogradely the cortical spinal neurons. And then we let the animal survive and we treat the animal with MPTP and then, sacrifice the animal and process the tissue. So we had to put in place, that's something which is new for us. So we had not done that before. So we have been doing a lot of stereotaxic brain injections in monkeys, but we needed to develop a laminectomy procedure to be able to inject these monkeys in the spinal cord. And we are doing that in collaboration with a team of neurosurgeon here, Nick Bullis and Thijs Bachman, who are shown here, who are part of our neurosurgery group here at Emory. And Nick particularly is really the spinal cord neurosurgeon. He does a lot of ALS work in human and pain, essentially. So been specialized himself in spinal cord surgery. So they have been working with us in developing the approach to be able to selectively target the cervical and thoracic spinal cord in these monkeys. So the way that we do that is first we take an X-ray and we put some little staples here on the top of the back of the animal so that we can locate the exact location of the vertebrae that we want to remove, which are mainly the lower cervical and upper thoracic that we are aiming for, because this is the area that is mainly related to the arm region. Then once that is done, and the animal in the surgery room here is fixed into this frame that Nick has developed, and so we put the animal into this frame in that prone position on the surgical table. And then, the laminectomy can proceed, and we can then have access to the spinal cord at the level of the cervical and thoracic spinal cord. And you can see here, this is a segment of the cord that we usually go to. That's the rostral part of the spinal cord here, the more caudal part, and then this is then the dura is put aside so that we can go through this and proceed with the injection of the viral vector into different parts of the spinal cord. Here, just to show you a little, let me just a second here. Just need to, yeah. Okay. So here's a little video of when this injection go on. So you can see the little tube here. So this is connected to the pump, and this is where the virus is coming from. And this tube is kind of floating, so that when even the spinal cord is moving, as you can see here, the needle is in the spinal cord and move with the cord, so that we don't make any lesion of the cord. So the way we proceed is we make series of injections on both sides of the spinal cord, and then that give us, this is the way that we proceed to deliver our viral vector into the cord. And then after that, really the dura is closed, and the rest is the skin as well, and the animal really will survive with that for many weeks. So following this injection, here's an example of one monkey that was injected with the virus. So this is this retrograde AAV that we are using here, which has the property of being efficiently transported in a retrograde fashion. So what we have done is to inject on both sides of the cord. On one side, the virus was marked with M. cherry, which is shown here. On the other side, we label it with GFP so that we can use essentially both sides of the cord and be able to differentiate which neuron go where. So, and here are what you see here are series of rostrocaudal series of section going from the front of the monkey brain, at least at the level of the striatum here, as you can see, caudate and putamen, going to the more kind of midbrain level, just to show you that the extent of labeling that we get into the cortex following this injection, which is pretty strong. So you get a large number of cells which invade mainly the M1, but also the SMA. And there is also neurons that are labeled in the more anterior region, in the premotor region as well, that I'm not showing here. But there is an extensive labeling of these cells throughout the motor cortices. And you can see that on both sides, either with both sides of the spinal cord, we got the same results. The other thing to notice as well is this, and we'll come back on that, is you can see as well that you see all this staining coming out of the motor cortex and going all the way down to the spinal cord, to the antennal capsule, and then the cerebral peduncle, and you will be able to follow that all the way to the spinal cord. So this is, something that this neuron, this virus, in addition to label the neuronal cell bodies extensively, also label the axon collaterals of these neurons. So we'll be able to take advantage of that to study the efferent connectome of these cells, and I'll be talking about that in a little bit. But you can see here that the extent, all those axons are really originating from these corticospinal neurons, and they're projecting and giving collateral to different subcortical region, and of course going at the end, going all the way down to the spinal cord. And here is just to show you an example of the kind of labeling that we get. And it is for the anatomical study, it was really important that we could access to a tracer that gave us, a full type of Golgi-like labeling of the neurons, since we wanted to study their morphology and wanted to study changes in the morphology of the dendritic tree. So we got that with this particular tracer. So you can see here that you see the very profuse basal dendrite of these neurons, but also the apical dendrites are labeled all the way from the layer 5 to the layer 1. And you can see that the dendrites are covered with spines that we'll be able to study and quantify and study in detail. And so that essentially, so this tracer, this virus allows us, to visualize these neurons into their entirety, going from the cell body, the dendrite, but also the axonal projection. So what is going on right now, taking advantage of this, then we are studying locally in the cortex. We're studying the morphology of this neuron using a variety of tools, mainly to the neurolucidus system. We're going to study the dendritic tree of these neurons, and also we're going to look at the volume of the dendritic tree, the changes in the number and complexity of the dendritic tree. And we're also looking at as I mentioned, at the density, the morphology of the dendritic spines on different segments of dendrite through the cortical mantle of these corticospinal neurons. And as I said, we're also doing that. So I showed you the spinal cord, but this tracer is also excellent to label other population of the corticofugal neurons. So we have animal that we have injected selectively into the medulla. So you can see here an injection into the lateral medulla, which also receive a significant input from the motor cortices, and you can see the extent of labeling into the motor cortex, and also the striatum, which of course we injected in the putamen here, and we collect data from the corticostriatal neuron. So by doing this, then we'll be able to somehow study and paying attention to these three population of cells. Of course, some of them are collaterals of each other. This is something that we want to study as well, but will be at least the prime. We'll try to primarily target them by injection into these specific targets, and start with a population that has been labeled by a very specific injection. So the question that come to mind there, in that we want to evolve also in terms of changes, is, are these neuroplastic changes in the morphology and the connections of corticofugal neuron linked to the development of Parkinsonian motor symptoms? Or are they happening early, as I mentioned at the beginning, during the process, and they are mainly homeostatic or pathologic changes, but happening during the course of nigrostriatal dopamine denervation? So this part, how we're going to do that is that, so these are different monkeys that these animals are going to be treated with MPTP, but instead of making them fully Parkinsonian, we're going to monitor the level of dopamine loss into the striatum using PET imaging, as you can see here. So what we use is a ligand for the dopamine transporter, which has been working really well in our study. So we published that, in many years ago now, but it's definitely a very efficient marker of the dopamine transporters. You can see the striatum here, and this is the immunostaining for TH and that in the striatum. And then this is an NPTP treated monkey. So we'll be using this to follow longitudinally the loss of dopamine in the striatum, and then we'll be euthanizing animals at different time point following the loss of dopamine. Some animal will be euthanized after about 30% loss of dopamine transporter ligand into the striatum, 50%, and then we will have the Parkinsonian animal, which really are over really 70% loss in the striatum. And we'll be looking at this, the same, some of that would be more what I just described before, looking at the morphology of the cell, but we're also interested in looking at the denervation of these, the cortical areas, both in glutamatergic, serotonergic, and catecholaminergic innervation of these regions in this monkey, because this chronic MPTP treated treatment affect not only the dopamine cell, but also affect the serotonin and the norepinephrine system. So that's the plan that we do to assess the changes during the course of dopamine denervation. This will be groups of different animals sacrificed at different time point, and we'll be able then to look at the changes going on in this population of neurons at different time point following dopamine denervation. So this was really the anatomy part of that we're doing. In parallel to that, as I told you, there's one slide here coming from, this is part of the group where, so Thomas Wickman and Adriano Galvin, in collaboration with Jonathan Nacy from in Scoppic in California. So they will be using the calcium imaging approaches to look at changes in activity of the population of corticospinal neurons, in a corticofugal neuron, essentially, in MPTP-treated animals. So you have a little bit here, an ideas of the type of, the studies are in progress, in fact, are just really starting, but you see a little bit about the design of their study here. So first of all, we'll take, MRI and a PET scan, a baseline PET scan for each monkey that undergo that. So the monkey are going to be trained to do a basic arm reach test. So all monkeys will be doing this. And then after that, the monkey will undergo surgery to label the neurons by injecting, again, the same virus as we talked before in the medulla, the striatum, and some of them maybe in the spinal cord. And then they will receive a second injection to induce the GCAMP into these population of neuron in the M1 and the SMA. And there will be the microendoscopy that will be implanted on the head of this animal to visualize the neuron. The neuron will be visual, the activity of cell will be visualized in the normal state. And then the animal, once they have collected data in the normal state, then the animal will undergo treatment with MPTP, and they'll be able to follow that longitudinally during the course of MPTP denervation. And the MPTP-induced dopamine denervation, again, the PET is going to be used to let them know about the state of the dopamine system as they collect, these imaging data. And then they will also look at the plan as this is to look at that in Parkinsonian state, and also the effect on this by using levodopa. So they'll be looking at different stages of dopamine denervation, and for the animal that will be Parkinsonian, then they will look at the effect of levodopa on the activity of these neurons. So that's a little bit about, that's one part really, which is really focused on what is going on in the cerebral cortex. And the other part is mainly is also related to these corticofugal neuron, but more focused on the efferent output of these cells. So as we know, and I think this has been well established for quite some time, there are changes going on following dopamine denervation into the cortical outflow. And there is evidence really that has been well established by many years ago by different groups. Clearly, we know that the corticostriatal system, for instance, becomes abnormal. So you have an hyper and overactivity of the corticostriatal glutamatergic system in animal model of Parkinson's disease. Here, this is data from our group in MPTP-treated monkeys. So you see there is an increase in the number of the GLUT1 terminals in the striatum of these monkeys. There are also ultrastructural changes in the corticostriatal synapses and spines. So you can see here that the in the MPTP in yellow, and control in the control monkey. The volume of the spine is increased. There are changes also in the postsynaptic density. The synapses are much bigger in Parkinsonian state, and the terminals also get larger. So there are morphological changes going on in the corticostriatal system in Parkinsonism. There's also changes going on in the subthalamic system in regard to the glutamatergic innervation. So there is loss of in that case, this is a loss of the VGlut1 terminal that is coming from the cerebral cortex. That has been shown in monkeys, in NPTP-treated monkeys by our group, but also been shown by Mark Bevan team at the time that, in fact, Ong-Yon Shu was a postdoc with Mark and them, and he's the one who kind of published that paper showing that this is also happening in 6-OHDA-treated mice, that you have this decreased glutamate terminals into the subthalamic nucleus of Parkinsonian animal. And Mark also, they also demonstrate in their case that there was also changes in the morphology of the GABAergic synapses coming from the globus pallidus into the STN of Parkinsonian mice. So there are multiple changes going on in the morphology, but also function of those input in the state of Parkinsonism. So the structural changes in synaptic connectivity contribute to the pathophysiology of the basic gain thalamocortical network in the Parkinsonian state. That has been the link of that. There's a lot of evidence suggesting that they are not only morphological, but they also have some important functional consequences. So whether or not, the question is whether or not these changes in the efferent connectivity affect a specific subpopulation of corticofugal neurons, and how the other, that's one question that we'd like to address. And also the other question is, that is more general in terms of the connectivity of corticofugal cell, is how can pyramidal tract corticofugal neuron be differentiated from each other by their pattern of axon collateralization? And that is something which is really important, which for reasons that are lack of tools to study that in great detail has been neglected, but clearly has been noticed, and is something important that we hope that we can advance really our understanding of that using this viral retrograde viral technique. And here's an idea, is an example of that, essentially to lay the foundation. So that's a nice review that Gordon Shepard from Northwestern published on the pyramidal tract and intratelencephalic neuron in the cortex in Nature Review Neuroscience within 2014. And so one point that Gordon was making in that review is that the pyramidal tract neuron in the cortex are in rodents are heavily collateralized. So in a sense that, you have the cell body in the cortex, but they give rise on their way to the spinal cord. These axons give rise to multiple collaterals to many, many different brain regions on the way down. So a heavily collateralized system. Okay, so this is something which is important. And this data, of course, was not necessarily only his data. So these data come from a nice paper that Hitoshi Kita did in 2012, where he used single cell thinning of cortical neuron and follow and their axon projection, and demonstrated that they were sending connection to practically a lot of brain region into before they reach the spinal cord. So a nice point that Gordon put into this, which I think is fundamental, is that in his review, so a clear but often overlooked implication of this is that behavioral functions of pyramidal tract neuron presumably derive from their output projection in toto, not just from a subset of branches. So making the point that we really need to be able to understand the functional role of these different output of the cortex, we really need to know where are they going, where do they go, what do they talk to, and that I think is clearly as important as knowing that this is a corticospinal neuron, because there might be different subtype of corticospinal neuron based on their connection with other subcortical regions. That was, I think, an important point that was made there. One thing that was a little bit kind of neglected in the review that Gordon, and we wrote a brief commentary following his review to make the point that really we know that, of course, these data were real and really nice data, and this was, and the rodent was clearly important. On the other hand, I think although the data were relatively sparse, there was not a lot of data, but the data that were available at that time in primates, and I mentioned those before by Rob Turner in particular and other data, were not really in line with this heavy collateralization. They were much more in line that there were really different subset of pyramidal neurons in the cortex of primate that were going to the striatum or going to the spinal cord, or maybe going to the subthalamic nucleus. But adding neurons that are collateralized as heavily as that was not something that there was any evidence in the primate brain that this was, existing. So we had wrote it with that, really, this little comments about try to make this point that maybe there is something going on here between rodents and primates in terms of axon collateralization. Of course, the problem was that really the data from the primate were really rare, were really sparse, and were really limited by technical issues. So as we mentioned, really there were some data coming from a single cell feeding in the cortex of primate that showed that was in line with that, but it was again a relatively small number of cells. There were this data from Rob Turner and other that was mainly antidromic invasion of cortical neurons, showing that corticostriatal and corticopontine neurons seems to be different. And also there were the data that were just shown you before that the firing changes in the corticopontine neuron is different than corticostriatal in Parkinson and monkeys. So all of those were in line that maybe there were different subset of neurons, but clearly they were limited by the sample size, and it was very difficult to make any clear conclusion about that. On the other hand, recently, just this year, in fact, this was this problem was approached in a different way by, in this paper here that was published in Yoland in 2022, in which they use essentially the approach that we are using more or less with different viruses, but the same idea, by injecting virus into the spinal cord, mainly the area of the cord that control the forelimb movement. Okay, they did that in rodents, and they did that also in primates. And then they compared the axonal projection of these corticospinal neurons through the brain of these animals following these injections. So that's really what they're showing here. So you have a figure here showing the labeling in the rat. This is rat that has been used here in the cortex of rat. So you see the retrogradely labeled cells in the layer 5 of the M1 region. And you see the same thing here in the primate in the arm region of the M1 of this. So the conclusion that they made here was, and here what you can see is the areas of the brain where they found axon collaterals following those injections. So as you can see here, the bar, there are many brain regions that contain significant, really, proportion of innervation density in the rat compared to the primate, which is really largely focused on the spinal cord. Okay, it's all spinal cord region here, and maybe an area here in the reticular formation. So and a little bit here in the thalamus, but mainly most of it is in the motor areas, essentially at the cord levels, with very limited collaterals to other brain regions. So what they conclude in that paper is that the corticospinal system in macaques, primates, essentially primarily constitute a final output system for fine motor control. So again, these are regions here coming from the arm area, the end area of the motor cortex, whereas this projection in rats exert a multimodal integrative role that accesses far broader CNS region, in particular sensory, so suggesting that there is much more sensory motor integration in the rodent at the subcortical level than there is in primates. So here we're taking advantage of this, using more or less a similar approach, and this is what I was, just to recall what we saw before. So this is the injection of this retrovirus in the cord that labeled those cells in the cortex, and then we can see the labeling of those collaterals throughout the brain. And here's an example of what we can see, for instance, in this animal that received the injection in the cord. When we look, for instance, here in the zona incerta, you can see this large field of terminals, which are terminals coming from collaterals of corticospinal neurons. You can also see some terminal in the subthalamic nucleus, which is the same thing. And then when you look in the striatum here, there is some, but it's relatively sparse, compared to other regions. But the bottom line is that we can do that through the different brain regions and be able to quantify and assess the extent of these innervation from different subset of cortical fugal neurons. And we'll be able to quantify that. So you can see here, we can use different methods that will allow to quantify this and compare that, between control and Parkinsonian monkeys, so that we can come up with some kind of schematic like this that will be able to make some densitometry measurement comparison between different brain regions and have an idea then about the connectome and the changes in the connectome of these neurons in the Parkinsonian state. Finally, the one thing that we'll need for that, of course, is we'll need to be able to quantify the cells. We need to be able to make some comparison between animals. We need to be able to count the number of neurons that are retrogradely labeled following those injections and the map of these neurons, where they are located, and doing this. So what we are doing for that, because of course that cannot be done with traditional and unbiased stereology method. These are too many areas to count this neuron, and it's too broad in scope. So what we have been doing over the past year is developing this relationship with this group, this Aphoria cloud platform, which is online, which essentially here is a artificial intelligence system, an algorithm that will allow us, we can build those algorithms to be able to count these neurons into the each monkey brain that we have. So we have been working with them, and we're developing those models that will allow us to automatically count the labeled cortical fugal neurons, in each monkey, and be able to provide us with large map of these neurons in terms of localization, but also of quantification. So I think you can show here, we have been using that for other approaches. Here is an example of the count of calbindin interneuron in the motor cortex of primate. So we have here, this is the way we annotate the cell, and then the system itself is going to count every neuron. Once the model has been established and you're satisfied with the model, you can then throw at this model, all the section that you want that the model has been trained with, and it will be able to give you all this data in a very short amount of time. So there's still some advantages to that. To other methods is that there is a lack of human bias. So once if the model is well established, then this is done really, and you don't have any bias by the human in the microscope to do this. And of course, it can be applicable to large brain areas. Okay, so then to conclude here, so there's no real major conclusion. I think, as I mentioned, I just wanted mainly to let you know what we are doing and what we're planning to do. But there's a few things based at least on the data that were published that before in different groups, is that there seem to be some changes going on. There is a breakdown of the thalamocortical projection to the deep cortical layers of M1 and SMA in these neurotoxin-based model of Parkinsonism. As I mentioned, it seems to be, we have seen that in MPTP monkeys, but it also seemed to happen in the 6-OHDA-treated rodent model of Parkinsonism. There's clearly also loss of spine on the subset of pyramidal tract cortical fugal neuron in M1 that has been demonstrated also in both species. So it seems to be something which is going across species. It's happening in the pathophysiology of that system. And the efferent connectome of the corticospinal neuron related to forelimb movement is far more collateralized in rodent than in primates. This is mainly so far the data, and I think our data goes in line with that as well. We have not yet, studied that, but our data are very similar to what has been published by this group using other type of viruses, but using a similar approach. And I think there seems to be this differential collateralization, in terms of motor outflow between rodents and primate, at least for areas that are controlling, fine movement. And one thing that, and mainly the time for our, first year of our Udall and this ASP has been mainly devoted to refining and developing and put in place technique and procedure that we can use for that. So we had to develop this laminectomy, which is working really well. I think we are able to move forward and generate and deliver that, into monkeys. Also the finding the virus has been a challenge. We have tried different AAVs to get to this result. So we found in our hands that this AAV2 retro is the one that works really well for us for labeling these neurons, allowing us to be able to visualize the cells, and also giving us the possibility of studying the connectome of these neurons because it labeled the axons as well. And finally, I think the development of the AI system to count these neurons is also going to be really critical for us to be able to advance the work and be able to provide detailed map of these neurons in terms of localization, but also quantification, and allows us to compare data between control and Parkinsonian monkeys. So I thank you very much, and I want, of course, really to thank the people that we have been working with. So we have a great team that we've been working together for years and years with me, and Thomas and I have been working for about 30 years together. And Adriana joined the team some time ago, so she's also a really great colleague. So Rosa Villalba, Jeff Parry, Susan Jenkins, Guna Singh are also great, working mainly on the anatomy part of the project. As I mentioned, Nick and Thijs are two neurosurgeons who help us with the laminectomy procedure. And here, this is the group, as I mentioned, this is the Emory and the people at the Van Andel Institute, who are part of our ASAP team. And of course, I thank very much the funding agencies, and I'd be happy to take any questions and suggestions you may have. Thank you very much.
Great. Thanks. I think we have time for a couple of questions. Does anybody want to go first before I ask a question or two? Okay. So let me ask a couple quick questions. So one thing is, you mentioned that Thomas and Adriana are going to be studying treated animals. I mean, as you know, there's some evidence that treatment affects synaptic remodeling, at least in the striatum. And you have all that nice stuff that you've done in cortex. Is there any plan to take a look at that as well? So
take a look at you mean in the striatum?
Well, no, in the cortex, to see if treatment would affect the kind of spine changes, synaptic remodeling.
Oh yeah, for sure. Yeah, that's why. So these the animal that Thomas and Adrian are going to use probably will not be, because of the penetration, the electrode and everything that was being put there, is probably going to be quite damaged. So it's not the ideal material to be able to study that in those animals. But that's why the process that we're using for generating the monkeys are exactly the same between the anatomical project and the functional project. So we hope that what we find into our anatomical work, is going to be an anatomical substrate for them to use to explain some of their functional data.
Absolutely. Okay. So just another question. You referred to this briefly at the very end of your talk, but I think, so the idea that there's a high collateralization of pyramidal tract neurons in primates, that goes along with, the idea that particularly involved in fine motor control and more innervation of anterior horn cells. But you're focusing really on it looks like motor M1. What about other regions that are, involved in less, say more midline or postural?
Yeah, you're right. So so that's a great point, Roger. I think, this is most likely, the idea would be that probably as you go away from the region of the brain that control the fine movement, like particularly because this is probably the finest really motor control, right? The finger movement in the primate, particularly with the very fine, and this is where you have also direct connection between M1 and the motor neuron per se in primates, and you don't have that in other species. So as you go away from that, in other regions that is like more really a medial musculature, or even really legs and so on, you probably lose a little bit of that. It's probably not as much. And this is something which is important that we have to pay attention to. And I had at some point discussion with Peter Strick as well. And it's clearly that's, I think it differs not. So I don't think we can take, any region of M1 and assume that this pattern is going to be the same. And definitely also these potential species, in terms of collateralization, most likely is not as pronounced when you go to those regions between rodents and primate as it is right now into this finger area. So that is something to pay attention to. You're right.
Yeah. Omar, you have a question?
Yeah, that was great. And going back to the CMPF neurons and their loss compared to perhaps not as much of a loss in VA, VL, VM. Do we have like a molecular level understanding perhaps of what's underlying that preference?
Yeah, that's a good point about how these neurons die, not only of course in these neurotoxin based model, but also in Parkinson disease itself, right? What is the underlying substrate of that? We don't know, bottom line, we don't know. But one thing which is particular about it, and I discussed that at some point with Jim Swammeyer, and Jim did not seem to think that this was one thing which is very particular about the CMPF neuron in the thalamus is that these neurons are at least, all these neurons are devoid of calbindin or calcium binding protein, right? They are completely devoid. If you stain the thalamus for calbindin, you have a big white spot in the middle. It will be the CMPF. All the other neurons is not. So that's one thing. On the other hand, some of them contain parvalbumin. They contain. It's not like they're all devoid of that, but those that contain parvalbumin, and at least in human, we have not looked at that. But in human, Glenda Holiday had a paper and she quantified those, and apparently she found that some that contained parvalbumin were relatively spared. They were not as affected as the one that were not, because it's not all neuron, right? You have seen it's about really 40, 50% of cell. So there is a population that is not affected by this. And so I've always thought that personally, maybe there is something related to calcium handling that may contribute to that. One thing that we are doing right now, we have done recently, which is also interesting, is that we are looking at the CMPF, but in terms of the using 3D reconstructions of individual neuron in the CMPF, and also looking at the mitochondria and changes. And we have found that clearly there is, even in some dendrite that are remaining there, we found some differences in terms of the morphology of the mitochondria between the CM neuron and the neuron in the VAVL, for instance. In terms of the mitochondria, the cristae of the mitochondria are really damaged in the CM neuron. And these are not like that in other. So all of this goes together that there is some form of neurotoxic event might be going on. How it is, in rodent, you can kill them by, if you put MPP plus in the striatum, you're going to kill those cells. So MPP plus, you're going to kill selectively PF neuron without affecting the cortical neuron, or so there's something related to the MPP plus toxicity, calcium, mitochondria changes that affect that. And most likely some of that might also contribute to their death in Parkinson. It could be, but it's not clear.
Oh, great.
The other interesting thing about that, Omar, though, is that there's not a lot of synuclein deposition in humans.
That's right.
With this prominent neuronal loss. So this is an area where there appears to be a dissociation between synucleinopathy and neurodegeneration.
You're right.
And maybe possibly because we just haven't looked carefully at other regions because the synucleinopathy is so prominent. But anyway, Glenda Halliday and her team deserve a lot of credit for picking this out. It's quite interesting.
That's interesting. Yeah, so anyway, but that's the stage where that is, Omar. I think this is it's an interesting, but there's still some work to be done in trying to understand what is but I know that really there is some other group working on that. I know that Bernard Bernard Berlin has done some nice work on this, trying to understand the function of that. And he's looking at that too now. He's looking at lesioning these nuclei in different ways. So he will probably come up with some papers soon that will also help advancing, knowledge about that.
And is there perhaps also any relationship to the receptors that they express? A CM neuron versus a VA versus VL, for example?
That could be that. Yeah, you talk about glutamate receptors, right? Or what?
Glutamate,
Yeah.
dopamine, acetylcholine.
Yeah.
Any of because if all of those systems are being altered, then the triggering processes in Parkinson's disease itself could be runaway excitation from loss or abnormal gain of some synapses.
Yeah.
No,
I agree. I think you need to look at different potential sources of that, but that's a good point. Yeah. Very good.
Okay. Any other questions for Yoland, or that was covered a lot of ground? Okay.
Good.
Well, Yoland, thank you very much. That was a really
my pleasure. So thanks for
fun.
the invitation. It was great really seeing you again, and thanks a
Yeah.
lot. And if people have any really questions or point that they want to chat about, I'm happy to answer any question by email or anything.
Okay, great. Hope to see you in person next year, actually.
Yeah.
Okay.
Take care. Bye-bye.
Take care. I
Bye.
think the executive committee meetings